প্রিয় ছাত্রীরা, আজকে মলিকুলার বায়োলজির ফিজিক্যাল ম্যাপিং অ্যান্ড সিকোয়েন্সিং অফ জিনোম এই টপিকটা আলোচনা করব। ইট ইজ লেকচার 1, DNA fingerprinting. Alec Jeffries was a molecular geneticist. He was inventor of DNA printing. He worked at Department of Genetics, University of Leicester, UK. His original papers, Jeffries J. Wilson, V. and Thin SL 1985, hypervariable mini-satellite regions in human DNA. His another paper is individual specific fingerprints of human DNA. DNA fingerprint analysis. Mr. Chan's family consists of mom, dad, and 4 kids. The parents have one daughter and one son together. Another daughter is from the mother's previous marriage, and the other son is adopted. Here are the DNA analysis results. Which child is adopted? Why? Question 2. Which child is from the mother's previous marriage? Why? Number 3. Who are the own children of Mr. and Mrs. Chen? Use of RFLP in parentage. RFLP means restriction fragment length polymorphism. Matching of DNA fingerprints with parents. Actual RFLP results is not color coded. They all look the same in the southern blot. Percentage information is from the band's size or position. In this result we see S2 is adopted son, S1 is biological son, D2 is daughter from previous marriage of Mrs. Chen, D1 is biological daughter. অর্থাৎ S1 and D1 are biological children of Mr. and Mrs. Chen. Physical mapping techniques. Restriction mapping, fluorescent in situ hybridization, that is FISH, sequence tagged site STS mapping. Restriction mapping, which locates the relative positions of on a DNA molecule of the recognition sequence for restriction endonucleases. FISH, in which marker location are mapped by hybridizing a probe containing the marker to intact chromosomes. STS mapping, in which the positions of short sequences are mapped by PCR and or hybridization analysis of genome fragments. Physical mapping, a map generated by genetic techniques is rarely sufficient for directing the sequencing phase of a genome project. This is for 2 reasons. The resolution of a genetic map depends on the number of crossovers that have been scored. Genetic maps have limited accuracy. The basic methodology for restriction mapping. The simplest way to construct a restriction map is to compare the fragment size produced when a DNA molecule is digested with 2 different restriction enzymes that recognize different target sequences. এখানে আমরা একটা ডিএনএ স্টাইল দেখতে পাচ্ছি 4.9 KB এর ম্যাপিংটা করা হচ্ছে। এখানে 2টা এনজাইম ব্যবহার করা হয়েছে, রেস্ট্রিকশন এন্ডোনিউক্লিয়াস দ্যাট ইজ EcoR1 অ্যান্ড ব্যামিচন, অ্যান্ড নাম্বার 3 তে দেওয়া আছে যে রিঅ্যাকশনে EcoR1 প্লাস ব্যামিচন একসাথে ব্যবহার করা হয়েছে। তো এখানে 3টা ভিন্ন ধরনের বিক্রিয়া দেখা যাচ্ছে। তো বিক্রিয়ার ফলাফলগুলো এখানে ব্যাখ্যা করা হয়েছে, ইন্টারপ্রিটেশন অফ দ্য ডাবল রেস্ট্রিকশন, এবং লাস্টে যেটা দেখা যাচ্ছে যে প্রেডিক্টেড রেজাল্টস অফ এ পার্শিয়াল ব্যামিচন রেস্ট্রিকশন। If map 1 is correct, then the partial restriction products will include a fragment of 1.2 plus 0.7 equal to 1.9 KB. If map 2 is correct, then the partial restriction products will include a fragment of 2 plus 0.7 equals 2.7 kilobase. এখানে জিন ম্যাপ করা হয়েছে, আমরা জিন ম্যাপ দেখতে পাচ্ছি, এবং এর ফলাফলে যে ম্যাপ যে দুটো দেখলাম যার ক্যালকুলেশনটা হচ্ছে যে ম্যাপ 2 is correct here. Limitation of restriction mapping. Restriction mapping is more applicable to small rather than large molecules, with the upper limit for the technique depending on the frequency of the restriction sites in the molecule being mapped. In practice, if a DNA molecule is less than 50 kb in length, it is usually possible to construct an unambiguous restriction map for a selection of enzymes with 6 nucleotide recognition sequences. The limitations of restriction mapping can be eased slightly by choosing enzymes expected to have infrequent cut sites, that is rare cutter, in the target DNA molecule.